Can everybody see my screen?
I can see it.
Okay, so we're going to do a quick review over the chapter and then move into some example questions that you might see relating to this chapter on the exam. And if you haven't already, do make sure to read over the chapter, skim over it to see if there's something that you don't know. Okay. I'm gonna skip over everything that you kind of already probably know from honors or honors biology or 8th grade chemistry. And this diagram is just showing that the molecules in the human body are highly selective. You can see how little hydrogen there are in the Earth's crust compared to the human body. And there's a lot for the rest as well, kind of filtering out and taking from phosphorus and then other metals. And so for this event, you don't really need to know the kind of bonding chemistry that's being shown here. So I'm going to skip over all this. But you do need to know functional groups. So if you don't already know, hydroxyl groups or hydroxyl is just OH minus, and a hydroxyl group is just an OH. A carbonyl group is a C double bonded to an O. And you can see it can be at the end of a molecule in which there's also a hydrogen coming off the carbon to kind of complete that 4 valence, or it can be in the center of a molecule as shown with a ketone. And so you do need to know aldehydes and ketones, those specific names, as they do come up often. And they are going to be used for solution testing later, which is an integral part of cell biology. And the last group here in the figure is their carboxyl group, which is just a C double bond to an O plus an OH. And also it's just the ionized form on the right. And so these groups are kind of relevant for proteins and nucleic acids. So amino groups are NH2, or they can be NH3 plus when they're in their ionized form. Phosphate groups are PO4 2 minus. And you can see here in the figure that they do show it's attached to an adenosine. And this one would be adenosine triphosphate because there are 3 phosphates. And then methyl groups are CH3, and they're typically attached during a process called methylation. And so there are a variety of compounds that are methylated. So I'm gonna just ask a question. So Daniel, what level of science are you at?
Can't hear you if you're speaking, by the way. Yeah, I said honors chem in the chat, by the way.
Okay. Have you taken honors biology? Okay, so I'm gonna skip over this as well. Okay, I don't know if you've forgotten as well, but here's just the general format for a fatty acid. So it's going to be a carboxyl head with a lot of CH2 groups. And so that's going to generate a lot of hydrophobicity. And it's going to come into play later. And so because of that kind of format, you can have kinks in your fatty acids, and that's going to come into relevance when you start discussing cell membranes and other components of movement, because these types of unsaturated fatty acids give gaps that allow for more movement and flexibility. And in a phospholipid, one of those positions is replaced with a phosphate group. And typically with phospholipids, you do see a polar group. And there are a variety of different polar groups that they can be on phospholipids. And later when we discuss apoptosis and some other processes, we're going to look into that in more depth. Okay, and this just shows the general format of a non-ionized amino acid. And so you can see there's an amino group and a carboxyl group, and there's an alpha carbon in the center and a hydrogen. And then coming out of the carbon can be any side chain. There are typically a certain number that you normally see around 20. Does anybody know, do either of you know what, sorry, one second. I'm gonna wait for Emily to join really quickly. Does anybody know what this ionized form of an amino acid is called? Emily or Chime, do you remember?
Sorry, wait. I had to rewind my headset for a second. Can you repeat the question?
Yeah. So what is the ionized form of an amino acid called?
Zwitterion.
Yeah. And that's kind of something you have to memorize, and it probably will show up at some point.
Yeah.
And so this is kind of just showing how the process of the sorry, a condensation between the carboxyl group and the amine group. And so you kind of get this peptide bond that's characterized by a CO and an NH. And this is just showing like a variety of linkages in a row. So you can see that there's a repeating pattern of this like peptide linkage, which is an O double bonded to a C, connected to an N, single bonded to an H. Does anybody want to tell me what they think the characteristics of PHE are?
Yeah, I could go ahead and answer. So since it doesn't have any functional groups, like hydroxide, which are usually polar, it's non like the properties of this would be that it's non-polar, and then like aromatic, or it's an aromatic groups. Like if you see a ring, it's pretty much always gonna be non-polar and hydrophobic.
Yeah. Daniel, what do you think lysine is? The LYS.
Like hydrophobic versus hydrophilic, right? That's what you're asking, right?
Yeah. Okay. You said, yep, hydrophilic. And you can kind of see with this here, there's an NH3, but then there's a plus. And so since positively charged, it's going to be polar. And so it is going to be hydrophilic. Okay. And it's just kind of showing the general format for a nucleic acid. So you're going to have a triphosphate, and then, sorry, this is for ATP. And so you're going to have a triphosphate, a ribose group, an adenine. And so there's like the ribose and adenine together are called adenosine. And that's where the name adenosine triphosphate comes from. And it's not always necessarily a triphosphate. You can see a diphosphate or even one phosphate. And so, and you'll see later when we get to glycolysis, how that whole process works in the cell. Okay, I just want to quickly get to the questions. And then this slide is just talking about how proteins could have many conformations. However, one of them is the most stable, and that has like two reasons, kind of like a thermodynamic reason, and also like the functional groups, the way they're arranged, also play into the conformations. And there are questions asking like what two factors are involved in deciding the shape of a protein. So you would just answer with those. And this slide is just talking about how like there's certain bondings that allow molecules to pair together better than others. And like if they're a poor match in terms of their like, for example, like polarities, like in an active site, an amino acid has side groups that interact with the enzyme. And so some of the time, like if it's a bad fit, it's not going to actually catalyze any reaction. Okay. So now we're going to get into the actual questions. So does anybody want to tell me what number 1 is?
Glucose.
Yeah. Number 2.
I'd be amylase because it digests starch, right?
That's right. And so also just to clarify, this all isn't in the chapter, but I'm just like getting questions so like you to understand how they're going to be applied. And so you probably have learned most of these in honors biology, but it's good to just review them as well.
Wait, so for fructose is, oh, fructose is a pentose. That's like the way to recognize it.
It is a pentose. And then galactose, I believe,
Yeah, that'd be
like
tough,
one
but
flipped. Yeah.
which one?
Galactose, for example, I think it forms beta linkages.
Oh,
Like,
oh, you mean the linkages themselves? Okay, okay.
yeah,
I was wondering like if there's any group that's flipped up versus down.
yeah, that'd be galactose.
Yeah. Okay.
Okay. And does anybody know number 3? Yeah, that's right. Did you say 4?
Yeah.
Yes.
Okay.
Wait. I asked this before.
Yeah. Does anybody know number 4?
It's an anticodon. Also,
right.
wait, Daniel, are you taking honors bio right now, or have you like taken the course and already done with it? Oh, you already took it. Cool.
Okay. Try to like answer more of these. You can just like send the answers in the chat, even if like you can't speak. Number 5.
B.
That's right. So the rough endoplasmic reticulum contains ribosomes. And so it's possible that you could be finding this type of ribosomal activity in rough endoplasmic reticulum. And then number 6. Did anybody see anything?
Wait, what's being read? That's codon.
That's right. And so, like, if you want to review the 3 kind of colored things they have in this figure, will be the codon and what's on the tRNA, or like tRNA ribosomal transferase or whatever. That's going to be the anticodon. Okay. And so this is kind of more of an application of what they like vaguely mentioned this chapter. And if you take an honors bio, you should be able to answer these as well. So just try to think through them. So does anybody know number 20?
It'd be dsDNA, right? Double-stranded DNA.
Yeah, and you can tell that because A bonds to T, and so A equals T here, and C bonds to G in a double-stranded DNA. And so since both of those are true, it's probably a double-stranded DNA. And also, you know it's not going to be a double-stranded RNA because there's 0% uracil. And while there is thymine. Oh, and by the way, thymine can be present in RNA, it just won't be present in large quantities. Okay. Has anybody number 21?
Wait, yeah, maybe we could like it's like since I answered 20, maybe like Daniel can answer 21 and then Emily can answer 22.
Okay.
Like we can do that. And like if you have a question or if you don't know, just like you can ask for more information. You're like you can ask for a hint or something like that.
So I think Daniel's next. So you can like, if we can't hear you, you can just type it in the chat as well, what you think the answer is. That's right. And I like your thought process. Like first you looked and checked if there was uracil, and then checked if the ANT and CNG aren't equal. That's like a really good way to approach it. Okay. Sorry. My mouse disappeared. Okay. Number 22.
Double-stranded RNA
And what's your reasoning?
because it has uracil and then adenine and guanine match up and then thymine and cytosine and uracil match up.
Are you sure that cytosine binds to uracil and adenine binds to guanine?
Wait, you're right. Sorry. A2 uracil. Okay, so then it's single-stranded RNA.
That's right.
Okay.
Yeah,
Yeah,
that's kind of like we're tricking you with that.
Yeah, sorry.
yeah, I was thinking the same
Like,
thing.
Okay, and then number 23.
It's molecule 4. It can be either single or double-stranded DNA, right? Because you don't like, or I don't know. I guess
you're
my
right.
rationale.
No, you are right. It is E.
Yeah,
Just because, like,
you can't tell whether
yeah.
it's just a coincidence or if, yeah.
Okay. Does anybody know how to do number 24? Well, I think Daniel would be next. So Daniel, do you want to tell us how to do number 24? Yep, that's right. Okay. And so you took honors bio, so you won't like have seen this before, but this is like kind of a common question that Mr. Wilk is on like FRQs and AP bio. So let's just go through and each person can tell me what they would put for each part. So like next is Emily. So what would you answer for part I? And I is like this C double bond to O connected to an ionized oxygen.
I think it could form ionic bonds. Wait. Yes for hydrogen bonds, and then no for hydrophobic interactions.
Yeah. So this is the a carboxyl group just with it's ionized, so there's going to be no hydrogen there. And so since it does have a negative charge, it can form an ionic bond. And they also, because of the polarity, can form a hydrogen bond, but it won't be able to be involved in a hydrophobic interaction because it's always going to be polar or like charged, and so it won't be hydrophobic. Okay, Chinmay number 2.
Wait, so could this form ionic bonds? No, could not, because it's not an ion. Could it form hydrogen bonds? No, because it's or well, could it? No, because carbon doesn't have form polar bonds. Then
I
could
was
it
just looking
be?
at the NH2 group.
Oh crap, my bad. Wait, the NH2 group.
Yeah, the amine group.
Oh, wait, but yeah, so that's what I'm talking about, right?
Oh, you were mentioning the carbon.
Oh no, I'm saying it can't form a hydrogen. Oh, just so not like in the context of the molecule. Oh shoot.
Yeah,
Okay, yeah,
just the
yes. Oh, okay, yeah, it can because nitrogen forms polar bonds with hydrogen, and then no, it can't be part of a hydrophobic interaction because it's a polar molecule.
yeah. Okay, Daniel, number 3. Okay, while I'm like waiting for your answer, I'll just like ask Emily for number 4.
For number 4, okay, so this is hydrophobic, so it won't form ionic bonds or hydrogen bonds, but it will form hydrophobic interactions.
That's right. Just because I want to like move the meeting on, I'll just like say number 3. And so it's a methyl group, so it will form no ionic bonds, no hydrogen bonds, and it can be hydrophobic. It can form hydrophobic interactions. And just like so you know, those hydrophobic interactions are often, or most of the time, are pi stacking. And I don't really know the chemical basis, and you don't really need to know that either. Just know that that's the most common hydrophobic interaction. And that's like referencing the pi orbitals, I believe, in that hydrophobic side chain and among them. Okay, and this is like the part where we start going more into the amino acids. So, Chenmei, do you want to do the first species?
Yeah, sounds good. We complete the. Well, I'm not sure I understand the question completely. So complete a table indicating. So I just need to fill 1 or 2. Is that right?
Yep. So, which one of these groups of amino acids will bind and which one will not?
Oh, okay. So, wait, lysine and glutamine, they will not, because they're the same charge, positive, right? And then ASN. Oh wait, there's a structure right there. What am I doing? Oh yeah, never mind. Then lysine and glutamine do form an ionic bond, obviously, because they're opposite charged. Then ASN and serine, yeah, they'll form hydrogen bond. And leucine and valine will form hydrophobic interactions. So, yeah.
Okay. So, what's your final answer?
Oh wait, species 1 versus species 2. The drug binds. Oh yeah, so it's species 1.
Yeah. And then the other one will be species 2.
Yeah.
Okay. So, what specific amino acid prevents the ribosome from species 2 from binding drug 1? So, I'll go back. So, tell me, now that we know what species 2 is, which specific amino acids like stopping that binding?
Is it arginine?
Yeah. And the reason I do believe that is because like they're both positive, so they'll actually repel. And then for D, in terms of non-covalent interactions, so intermolecular forces, explain why the ribosome from species 2 will not bind drug 1, but the ribosome from E. coli species 1 will. So talk about like the IMFs at play right now in like this scenario. Or like which amino acids IMFs cause it not to bind versus to bind for the other species. Okay, yeah, it's just the glutamine and the lysine and the arginine and the lysine. I'm gonna just talk about how glutamine, or technically glutamate, has a carboxyl group, while lysine is positively charged with that a charged amino group. So you can talk about how there's an ionic interaction there. So that's non-covalent, but arginine and lysine won't have that interaction. Okay, I am running out of time, but I do recommend you look at this last question. And this is from like the previous questions were from like an easier level cell biology test, but these are from the MIT Biochem exams. I do think these are like more similar to the difficulty of like questions you would find for amino acids at like higher level science tip competition. So it's good to know this. And I don't think there's any reading to do for this meeting, just because I asked you guys to do chapter 3 last week. And so if you haven't done that yet, you can obviously just like skim over it. If you find something you don't know, just like write it down and send it to me. Otherwise, you don't have to take any notes. So it really shouldn't take that much time. And yeah, does anybody have any questions?
No, thank you.
Okay,
Yeah. Thanks. Thank you so much.
bye.